Section 4.3 is on DNA replication in the cell cycle. These are the objectives listed in the notes I've posted online. We'll look at them one at a time. So our first objective of this section is to describe how DNA is replicated. Now, before a cell divides into two new cells, we need to duplicate its DNA and make copies of it. That way each new cell gets exact copies. This duplication is going to follow the law of complementary base pairing, which we already know that A always pairs with T in a DNA molecule, double helix. And so what this law is talking about is that if we were to pull apart the strands and look at one of the strands, it's going to have a sequence of bases. Say a small segment here is A, T, G, A, T, A, and so on. But if we know what one strand looks like, then the base pairing rules will tell us what the other strand would look like. So basically we would need one strand to make the new strand or the new copy. And so that's kind of what this process will follow. So let's take a look at that.
DNA replication is the process in which one double-stranded DNA molecule is used to create two double-stranded molecules with identical DNA sequences. For this process to occur, the two DNA strands are separated and the DNA replication machinery is assembled at the replication fork. The two DNA strands are used as template strands to create newly synthesized DNA strands. The key feature that allows a DNA molecule to produce two DNA molecules with the same DNA sequence is the ATGC rule, also known as Chargaff's rule. According to this rule, adenine in one strand only hydrogen bonds with thymine, and guanine only hydrogen bonds with cytosine. For example, an incoming nucleotide with a cytosine base will only hydrogen bond with a guanine base in the template strand. By this mechanism, one double-stranded DNA molecule provides the information to create two double-stranded molecules with identical base sequences. Each copy contains one newly synthesized strand and one original strand. This mechanism is called semi-conservative DNA replication because half of the DNA molecule is conserved from the original molecule.
For DNA replication, there's 4 steps. First of all, we have to unwind the DNA from the histones, and then we got to unzip the DNA or separate the strands, and then we got to build new DNA strands, and then we have to repackage back with the histones. So first we're going to unwind DNA from its histones. Then we're going to unzip it with the help of helicase, which is an enzyme that will split apart the two strands. At the point where the DNA is open is going to be called a replication fork, and DNA polymerase will build the new DNA strands. So DNA is a big polymer, so that's where it gets the name polymerase. So this diagram here summarizes the process. So here is your DNA helicase, which is unzipping the two strands. Recall that the two strands are anti-parallel. The strand on top is going in this direction, is going 5 prime 3 prime. The other one is going in the opposite direction. So the other one is going 3 prime 5 prime. Okay, so the DNA molecule or the DNA helicase will separate the two strands. There are going to be some other proteins which you won't be responsible for on my exams, but these strands help to stabilize or keep the molecules apart. Otherwise, they'd want to come right back together. In the third step here, the leading strand, which is going to be the top strand, is going to be synthesized continuously in the 5 prime to 3 prime direction by DNA polymerase. The thing about this DNA polymerase is it can only add new nucleotides to the free 3' end. Okay? So because this strand would be made in that direction where there is always going to be a free 3', it can just keep adding continuously as the helicase continues to move in this direction over here in our diagram to the left and opening up the DNA molecule further. This DNA polymerase can just follow along. The other strand, though, goes in the opposite direction, so this is going to be called the lagging strand here. And it's going to be synthesized discontinuously. Here the primase synthesizes a short RNA primer. So here's our short RNA primer, and there's primase. As the replication fork moves further down the DNA molecule, it'll open up more space so that this primase can go and add a primer. From that primer there, then the DNA polymerase can attach on and add a segment of DNA. These segments are going to be called Okazaki fragments. So this is done a little at a time because, again, the DNA polymerase can only add to the free 3' end. So this lagging strand is going to be made in pieces as the helicase over here continues to open in our diagram to the left. So these Okazaki fragments are made. Then we have to knock off the primer, and in step 5 here, another enzyme that you're going to be responsible for, DNA ligase, is going to connect the segments together on there. So the DNA ligase is responsible for joining those Okazaki fragments. At the bottom of this diagram, this is the overall direction of replication. But if we followed to the other side of this, there's going to be another replication fork over here. And so the same thing is happening in this, going in this direction on the opposite side, on the other replication fork, except things are going to be reversed from top. What's going on top will be on the bottom, and the bottom on top. So because of the directions that the DNA polymerase can only attach to the new nucleotides to the free 3' end. So let's look at this from that previous diagram. What are some of the details you need to know regarding the adding of or building of the new DNA strands? First of all, polymerase is going to read exposed bases and match complementary free nucleotides using that law of complementary base pairing. Separate polymerase molecules are going to work on each strand, proceeding in opposite directions. Remember, one is going to be the lagging strand, the other one's going to be the leading strand. The polymerase moving towards the replication fork is going to make a long, continuous new strand of DNA, whereas the polymerase that moves away from the replication fork makes short segments of DNA. And then DNA ligase will come in and join those segments on the lagging strand. Ultimately, you're going to have two daughter DNA molecules that were made from the original parent DNA. So if you looked at the two daughter DNA molecules, each one is going to be made of one old strand and one new strand, and we're going to call that semi-conservative replication. And semi-conservative just means that, well, one of the strands in the new DNA molecules is from the old DNA molecule.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule. The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the two complementary strands of the DNA molecule are oriented in opposite directions and the DNA polymerase can only accommodate replication in one direction, two different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
And then the fourth step where newly made DNA is going to be repackaged. During this replication process, there's going to be thousands of polymerase molecules working on the DNA within that cell. And all 46 chromosomes are going to be replicated within 6 to 8 hours. During this replication process, histones are being made in the cytoplasm, and then they'll be transported into the nucleus soon after the replication ends. So then each of the new DNA molecules will now rewrap around the histones, forming those nucleosomes that we studied when we were looking at the structure of DNA and how it wraps up into chromosomes. This is a diagram from your book showing that process again. We already went through it on the previous diagram, but you can see here, this is just a representation of the helicase right here. The strand on top would be the leading strand. You only have one DNA polymerase, and it's going to be following the replication fork. Here you're going to have multiple DNA polymerases making segments in the lagging strand. And in this model here, this diagram, the one with the gold strand representing the sugar phosphate backbone, you can see that on both ends would represent the new strand of DNA being made, whereas the blue, symbolized blue, sugar phosphate backbone strand would be the old strand. So here, this is one of our new DNA molecules. We have the other one being formed. Ultimately, when the helicase gets to the end here and the process is ended, you're going to have two new strands, and each one is one old strand and one new strand, or semi-conservative replication. Next objective is to discuss the consequences of replication errors. So the DNA polymerase can make mistakes, but there's multiple ways of correcting those replication errors. The DNA polymerase double checks and replaces incorrect, unstable pairs with the more stable ones. So DNA kind of self-corrects. The end result is we have one error per billion base pairs replicated. So that's kind of remarkable. And so there's mechanisms for fixing those errors. Every now and then, though, one does get passed. And so that's where we get mutations. So mutations are going to be changes in DNA structure due to replication errors or environmental factors. So you could have agents in the environment like radiation or viruses or chemicals that can cause changes to the DNA structure. And some of these mutations can have no ill effects at all. In fact, some may provide advantages. Get back to the slide there. Some may provide advantages should the environment change, but at the moment that that error has occurred, it may give no advantage at some point. So it could lead to an adaptation later, even though it's sort of a neutral. Other mutations, though, can be bad, and they can kill. Others can cause cells to become cancerous, and others can cause genetic disorders in future generations. So these are the consequences of mutations. Some are neutral with no apparent consequence. Some are lethal, and some can cause genetic disorders. Our third objective for this section is to describe the life history of the cell, which includes the events of mitosis. So we have here a diagram we've seen in the lab before. It shows the cell cycle there and the stages the cell goes through. So the cell cycle, this is the cell's life from one division to the next, and it includes interphase and a mitotic phase. So on this diagram, the area indicated by the green is interphase, and the area up here at the top is the mitotic phase, or the M phase, the phase where division is taking place. Interphase is going to have three subphases. There's going to be G1 or gap 1, S, and G2. The mitotic phase includes its subphases, and those include prophase, metaphase, anaphase, telophase. And then not part of actual mitosis, which is the division of the nucleus, here we have cytokinesis, in which the entire cell divides. So during G1, which is the first gap phase, this is the interval between cell birth and DNA replication. So when the cells divide, the cell's going to grow. It's going to carry out its normal tasks and accumulate materials for the next phase. The next phase is S phase, which stands for synthesis. And here's where DNA replication occurs. And the centrioles are also going to be duplicated at that point, where the centrioles are involved in organizing the spindle fiber that pull chromosomes apart. And then G2 phase, this is the second gap phase, and this is interval between DNA replication and actual cell division. Here, the cell repairs any replication errors, and then it's going to grow a bit further, and it's going to synthesize enzymes that are involved in cell division. And then we have the mitotic phase, or the M phase. Here, it says the cell replicates its nucleus, but that's another way of saying the cell is going to divide its nucleus, which is what mitosis stands for. And then finally, we're going to have the cell pinch off altogether during cytokinesis. Some cells don't keep going through divisions. They are going to be or stop in G1. And so they never divide again. And so this would be called a G0 phase. And this describes a cell that left the cell cycle and ceases to divide for a long time. Examples would be like muscle cells and nervous cells. For other cells, the duration will vary, and it depends on the cell types that we find in there. Mitosis then is the division of a cell, and this results in two genetically identical daughter cells, and that's a key thing there, so I underlined it, because there's another kind of division called meiosis that produces sex cells and like sperm and egg cells, and those cells are not identical to each other. Every sperm cell produced, every egg cell is going to be different slightly, which explains why even brothers and sisters look different from each other. But what are the functions of mitosis? The function is in the development of an individual from the fertilized egg, or from one cell, to making 50 trillion cells that make up your body. Mitosis is responsible for the growth of all tissues and organs after birth. Mitosis is responsible for replacement of cells when they die, and it's also responsible for repairing damaged tissues. There's 4 phases of mitosis. There's prophase, metaphase, anaphase, and telophase. We have studied these in the lab already, so this will probably be mostly a review. The details of these are displayed on the following pages and on the notes that you are able to download for class. So I'm not going to really read through all of this here. Instead, I'm going to go straight to the diagram. So we're going to look at prophase. So right here, during prophase, you can see that you can now see chromosomes, because prior to prophase, during interphase, they were decondensed, and they're in the form of chromatin, which is too thin to be seen by a light microscope. So here, the chromosomes are condensed. You can now see them. The nuclear envelope is starting to break apart right here. Spindle fibers, these fibers right here, are starting to grow. And the centrioles are starting to migrate to opposite poles. So one centriole will end up on one side, another one will end up on the other side. The picture on the right here just shows a picture of an actual animal cell going through prophase. In metaphase, right here, the chromosomes lie along the midline. Sometimes we call it the metaphase plate. Some spindle fibers are going to attach to the kinetochores. Those kinetochores are located on opposite sides of the chromosome, where the centromere is. And the fibers of aster are going to attach to the plasma membrane. So these fibers right here, the shorter fibers, are going to attach to the plasma membrane to help support the spindle fiber structure. And then anaphase is described in telophase on these pages of the notes. But going to the diagram, which summarizes what's going on here. During anaphase, the centromeres are going to divide in two. So the centromeres divide in two, so basically the sister chromatids are split apart. The spindle fibers are going to pull the sister chromatids to opposite poles. And so now each side, when they get to the opposite sides, are going to have complete copies of those chromosomes. And then we have telophase over here. Telophase is kind of like the opposite of what happens during prophase. The chromosomes are now separated. They were once called chromatids, we now call them chromosomes. You can see the nuclear envelope is reforming, and the chromosomes are going to start to decondense. Eventually, the new nucleolus or new nucleoli are going to appear, and the cell will pinch off in the middle. So this structure is called the cleavage furrow, which is what's happening during cytokinesis. So during cytokinesis, we'll pinch off and completely form two new cells. So here's a definition of cytokinesis: is division of cytoplasm into two cells. It's actually not considered technically part of mitosis itself. The cytokinesis is going to occur during, or begin to start happening during telophase. So when the nucleus is dividing and you're having two new complete nucleus forming, you also have cytokinesis occurring at the same time. Now, how does this pinching off of the cell occur? It happens with the help of proteins that you actually find in your muscles called myosin and actin. So they start to constrict as they shorten. These fibers will begin to shorten and constrict and pinch off the middle. As it begins to pinch, it forms the cleavage furrow, which we saw on the previous slide, around the equator, and eventually pinching off the cells into two new cells. So what about the timing? How does a cell know when to divide? How does it know when not to divide? Well, some observations have been made that the cell is going to divide when the cell has enough cytoplasm for two daughter cells. If it's not big enough, it won't divide. It's going to divide when the DNA is replicated. You can't get two new cells without copies of all the instructions. The cells have to have, or the dividing cell would have to have, an adequate supply of nutrition. And then there's going to be chemical messengers or messages or growth factors, chemical signals that would stimulate the cell to begin division. And neighboring cells might die and open up space. So the dying of a cell nearby would be responsible for also helping to tell the neighboring cells to begin to go through mitosis. What about stopping division? The cells are going to stop dividing when they snugly fit up against neighboring cells. And nutrients or growth factors are withdrawn, so if there's not enough nutrients or you don't have the growth factors, then the cell won't go through division. And the cells undergo contact inhibition, which is the stopping of a cell division in response to contact with other cells.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into 3 discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.